Hello, this is Dr. Mathangi, Associate Professor of Biochemistry. In today's session, we are going to learn about hemoglobin, its metabolism, variants of hemoglobin, abnormal hemoglobin, and the diseases associated with it. So, how does a doctor assess the glycemic status of an individual with the HbA1c percentage? So, this chart explains the correlation between the plasma sugar level and the HbA1c percentage. Now, if there is any discrepancy between the plasma level and the HbA1c percentage are mostly relied upon in comparison with the plasma blood sugar level, as the plasma blood sugar level are bound to have fluctuations depending upon the dietary habit of an individual or the medication taken by an individual before the test. Whereas HbA1c is considered a gold standard in biochemistry for the assessment of glycemic status, as the values do not change nearly for a period of 3 months. So, what are the factors which would interfere with the HbA1c levels? So, the following conditions can give a false high HbA1c values. The condition being iron deficiency anemia, thalassemia, hypertriglyceridemia, consumption of certain protease inhibitors and immunosuppressants will elevate the HbA1c levels to a greater extent than normal. Also, there are certain conditions which will give a false low or negative level for HbA1c, and that being pregnancy, hemorrhage, iron supplementation, alcoholism and cirrhosis. Therefore, HbA1c level, though it is considered a gold standard, it is not the only thing for monitoring the glycemic status of an individual. Always it has to be accompanied with a fasting blood sugar level or OGTT level for better understanding of the glycemic status of an individual. Moving on to the next one, abnormal variants of hemoglobin. So, we have seen the normal variants as HbA1, A2, fetal hemoglobin, embryonic hemoglobin, and HbA1c, and its increased level in case of diabetes mellitus. Now, moving on to abnormal variants. Abnormal variants of hemoglobin are hemoglobin S, which is seen in sickle cell anemia, and thalassemia, where again the synthesis of the hemoglobin chains, either alpha or beta, is defective. So, first to start with sickle cell anemia. Sickle cell anemia, the hemoglobin molecule becomes abnormal, and it is not called as hemoglobin A, instead called as hemoglobin S for sickling. It is an autosomal recessive trait which is inherited from parent to offspring. Of course, the homozygous individuals who inherit the recessive mutant allele from both the parents suffer from the disease, whereas the heterozygous are carriers. Of course, they will have day-to-day abnormalities, however, it is not life-threatening. So, this autosomal recessive trait is because of a single point mutation in chromosome number 11. There are two types of sickle cell anemia. One is sickle cell anemia, where, as I told you already, where an individual has inherited two recessive autosomal gene from the parents and he is termed homozygous. And the second condition being sickle cell trait, which is actually a milder form, where only one mutant allele is inherited from any one of the parent, and the state is said to be heterozygous. So, the point mutation occurs in chromosome number 11 that codes for the beta chain. Specifically, the amino acid glutamate is replaced by the amino acid valine. So, this diagram, it shows the single point mutation, meaning single nucleotide alteration in the DNA, where adenine is replaced by thymine. Subsequently, it brings about the change in the codon of mRNA, where GAG, which codes for the normal protein glutamate, becomes GUG, which codes for valine, and the protein becomes mutant. The protein here we are talking about is the beta chain of human adult hemoglobin, which has 2 alpha chains and 2 beta chains. In the beta chain, as we have seen already, among the 140 plus amino acids, just the 6th amino acid is replaced, that is glutamate is replaced with valine, no other alterations, but this brings a huge change in the shape of the hemoglobin and also the shape of the RBC. The RBC, which is normally biconcave disc shaped, now it assumes a sickle shape because of this point mutation and change in one amino acid. So, what are the consequences of sickle cell anemia? Because of the sickling of the RBC, there is ineffective transport of oxygen, and also the sickled RBC could clog the capillaries, affecting major organ system of the body. Normally, patients with the sickle cell anemia will be called the sufferers, as they have a severe anemia and inability to transport oxygen. Whereas the sickle cell trait patients, they will be mildly anemic and they will have deficient oxygen supply upon strenuous muscular exercise or upon reaching a higher altitude. Otherwise, the day-to-day life would be normal. Moving on to the next abnormal derivative of hemoglobin, that is called as thalassemia. This is also called as a quantitative gene effect. There are two types of thalassemia, thalassemia type alpha and type beta. Alpha thalassemia, it results because of abnormal synthesis of the alpha chain of the hemoglobin. Here the mutation is with chromosome 16, which codes for the alpha chain, and there are two genes that are mutated, HbA1 and HbA2. So beta thalassemia occurs because of the defective synthesis of the beta chain of the hemoglobin. Here the mutation is associated with chromosome 11 and the defective gene is hemoglobin B or HBB. So alpha thalassemia is classified into alpha thalassemia major and minor. The same way beta thalassemia is also classified as beta thalassemia major and minor. As the name indicates, beta thalassemia major is life threatening and the person's life expectancy is bound to be around 35 years, and normally individuals don't survive beyond the age of 35. So going to alpha thalassemia minor, those who have inherited the defective gene of alpha chain, they do not show any symptom. That is because the alpha gene defect doesn't cause any major problem, or it does not interfere with the normal functioning of the hemoglobin. Whereas persons who have beta chain of hemoglobin that is defective may have mild anemia. Beta thalassemia major is normally called as hemoglobin H or Cooley's anemia, and this here the symptoms are very severe and it is very evident also in the within the first 2 years of life, and this delays growth, causes infection, enlargement of liver, spleen and heart. So we have seen the normal variants, abnormal variants and the derivatives of hemoglobin. Now we move on to the next topic, that is methemoglobin. This again is of a greater significance in exam point of view. What is methemoglobin? Methemoglobin is again an abnormal derivative of hemoglobin, which results because of the oxidation of the iron present in the heme moiety from Fe2 plus state to Fe3 plus state. So this is very dangerous because the oxidation is not reversible, and Fe3 plus in heme in Fe3 plus state cannot bind oxygen, therefore it becomes useless. So Fe3 plus, oxidation of Fe2 plus to Fe3 plus happens normally in cyanide poisoning, where the cyanide ions which are negatively charged bind to the Fe3 plus and it forms cyanomethemoglobin. Methemoglobinemia is also common in individuals with the glucose 6-phosphate dehydrogenase deficiency. So moving to the next molecule, myoglobin. So myo means muscle and globin is a protein. Myoglobin is a type of protein which is exclusively present in the muscle, and it helps to deliver oxygen in the absence of enough supply of oxygen to the tissue. So normally it is present in skeletal muscle. Unlike hemoglobin, myoglobin has a single chain of polypeptide and a single heme unit. Therefore, one molecule of myoglobin can bind with only one oxygen molecule. However, the binding of myoglobin to oxygen is much higher. Therefore, the dissociation of oxygen from myoglobin does not happen unless the partial pressure of oxygen in the tissue falls below 40 mmHg. The excretion of myoglobin in urine is considered as a marker for various disease, and the condition is termed as myoglobinuria. The fluctuations in the level of myoglobin or its presence in plasma is considered as a biomarker for myocardial infarction. The structure of myoglobin is shown. It is very much similar to that of hemoglobin, but it has a single chain of polypeptide. Moving on to the next one, heme containing proteins and enzymes. Other than hemoglobin and myoglobin, there are also other proteins which are present in the human body that contains the metalloporphyrin ring with the iron atom, which we call it as heme as its prosthetic group. Just get familiarized with the names of those heme containing proteins and enzymes other than hemoglobin and myoglobin. So the following are those hemoproteins: cytochromes. Cytochromes plays a significant role in electron transport chain. The second one is catalases. Catalases are the ones which helps in the decomposition of hydrogen peroxide. They play a significant role in free radical scavenging. Peroxidases in general also plays a significant role in free radical scavenging and also are widely expressed in white blood cells, which helps in phagocytosis. And the most important enzyme which contains heme is tryptophan pyrrolase. So, these are other heme containing proteins. They do not help in transport of oxygen. However, they help in functioning of various enzymes and they act as prosthetic group. Moving on to the efficiency of hemoglobin in transporting oxygen. So, this relationship is repeatedly asked in questions. So, this is called as hemoglobin dissociation curve. So, this curve is drawn to show the dissociation of hemoglobin from a single molecule of dissociation of oxygen from the single molecule of hemoglobin. As we have learnt now, hemoglobin binds with 4 molecules of oxygen, and I told you under normal condition it will not be able to dissociate to give all the 4 molecules of oxygen, because the binding is positive, it is cooperative, and the affinity of binding is much higher. So, the binding forces have to be overcome to break the oxygen molecule and to release it to the tissue. So, when we plot a graph between the partial pressure of oxygen in the tissue or cell with respect to the percentage saturation, we normally expect a linear curve, meaning when there is a decrease in the partial pressure of oxygen in the tissue, hemoglobin should dissociate to give oxygen. However, in practicality it is not so. The curve is not linear and it is sigmoidal. That is, even when the partial pressure of oxygen in tissue or cell is 40 mmHg, hemoglobin will be 50% saturated. That means it would have dissociated to give only 2 molecules of oxygen out of 4. And also remember, the minimum partial pressure of oxygen in any living cell would be 40. So, under that condition, hemoglobin dissociates only 50% and it is not 100%. So, when we plot a graph, we get a S-shaped curve called as a sigmoidal curve. So, when will the curve shift to the right? When the curve shifts to the right, it means that in addition the hemoglobin is delivering more oxygen to the tissue. So, the curve will shift to the right when there is an increase in temperature, body temperature, as in the case of fever, when there is increased presence of 2,3-bisphosphoglycerate, which is an intermediate of glycolysis seen only in RBC, or when there is an increase in hydrogen ion concentration, or when there is a decrease in pH, which is normally correlated with severe metabolic activity, when more metabolic acids are produced. Under such condition, the curve will shift to the right and there will be increased dissociation of oxygen from the hemoglobin. On the other hand, when will the curve shift towards the left? The left shift is seen in conditions quite opposite to that of right shift. That is, when there is a decrease in temperature, it is hypothermia. When there is decreased 2,3-bisphosphoglycerate production, this occurs in lack of oxygen. And when there is a decrease in H+, meaning increase in pH, and in carbon monoxide poisoning, the curve will shift towards the left. So, this is called as Bohr's hemoglobin or oxygen dissociation curve, and always remember the shape of the curve is sigmoidal. The shift to the right means easy dissociation of oxygen from hemoglobin, and the shift to the left means there is decreased oxygen dissociation from hemoglobin. So, moving on to the next one, chloride hamburger shift. So, this relationship is much important to understand the transport of carbon dioxide between RBC and plasma. So, carbon dioxide from the tissue enters the RBC. It binds with water molecule in the catalyzed by the enzyme carbonic anhydrase, gets converted into carbonic acid. So, this is an enzyme catalyzed reaction. The carbonic acid is a weak acid and it dissociates to give bicarbonate ion and H+ ion. This H+, as I told you already, will be bound with the free hemoglobin or the deoxygenated hemoglobin and it forms HHB. So, this bicarbonate ion which gets accumulated in the RBC has to be transported in the plasma, which has to reach the alveoli to reform the carbonic acid and gets split into carbon dioxide and water for exchange. So, when the bicarbonate ion move out of the RBC, it creates a disturbance in the electrolyte balance. As a result, chloride from the plasma enters the RBC to maintain the electrochemical neutrality, and this is called as chloride shift or chloride hamburger shift. The reverse of the chloride shift occurs in tissues. The reverse of the chloride shift will occur in tissue where chloride will be pushed outside into the plasma and bicarbonate will move in. So, this is an interesting concept which has to be remembered to understand how the oxygenated hemoglobin and deoxygenated hemoglobin function inside our body. This also helps in buffering the pH of blood, which will be dealt in the later chapters. So, let us recall the learning outcomes for today's module. We learnt about the structure of hemoglobin along with its property. We discussed what is tetrameric, what is allosteric, what is conjugate protein, and we also discussed the concept of positive cooperative binding. We understood the normal derivatives of hemoglobin which are present in human blood as HbA1, A2, E and F, along with the subunits, the alpha subunit remaining the same, whereas the other two subunits vary with a different type of hemoglobin. We also studied the abnormal variants of hemoglobin and the normal derivatives of hemoglobin. The normal derivatives of hemoglobin are carbaminohemoglobin, oxyhemoglobin. Abnormal derivatives are seen in carbon monoxide poisoning, it is carboxyhemoglobin. HbA1c is also a normal derivative of hemoglobin. However, when the percentage of HbA1c becomes more than 6.5%, it indicates diabetes. So, we also saw the genetic disorders associated with hemoglobin formation and metabolism, principally focusing on sickle cell anemia and thalassemia. Both are autosomal inherited as autosomal recessive trait, and in sickle cell anemia there is a point mutation affecting the beta chain synthesis. Whereas it is a regular gene mutation affecting either the synthesis of the alpha chain where two genes are affected or the beta chain where one gene is affected. Again, the pattern of inheritance is autosomal recessive. And then we saw about methemoglobin and certain proteins and enzymes which contain heme as their prosthetic group. In addition, we discussed the sigmoidal curve of hemoglobin binding with oxygen and the Bohr shift and the chloride hamburger shift. Kindly get back to us if you have any queries, and
this concludes my session. If you have any questions, come back.
This concludes the session on hemoglobin. If you have any doubts or queries, please write to us. We will be more than happy to help you. Thank you.